Welcome to Behind the Scenes Making an Intranasal Vaccine. In this episode, we are going to Groningen, the Netherlands. We are meeting with Prof Dr Anke Huckriede. Professor Huckriede is Professor of Vaccinology at the University Medical Center Groningen, Faculty Medical Sciences. Professor Huckriede's research focus is on influenza and COVID-19 vaccines.
We have been using influenza vaccines since about 50 years, I think, or even more than that. But in fact, we still use more or less what we had at that time. And there has been relatively little innovation, except for that an additional strain has been added to the vaccine. Originally, it contained 3 different influenza virus strains. Now there's a 4th one in it. But next to that, not much has changed. There have been many activities, though, in order to improve the current vaccines and also to get a longer duration of immunity, but especially to make a vaccine which covers a number of strains so that maybe not every time, every year we would need to get a new vaccination. So working towards a broadly protective vaccine or maybe even a universal influenza vaccine is something which is an active field of research. But there are now also some approaches which have proven to be quite effective already in clinical trials. And in my eyes, the most successful at this moment is an approach where the researchers try to focus the immune response to the conserved stalk part of the hemagglutinin molecule. So the molecule consists of a head region, which is rather variable among the different influenza virus strains, and a conserved stalk region. And by focusing the immune response on the conserved stalk regions, they were successful in inducing antibodies which recognize not only one strain of virus, but a number of different strains. How effective these antibodies would be to protect from influenza virus infection in the human context remains to be seen. That has not been yet investigated.
Is there also a focus on T-cell-based immunity and, for example, on T-cell-based vaccines?
T-cell-based vaccines are indeed quite interesting in the context of influenza because the T-cell epitopes, so the parts of the virus proteins recognized by T-cells, these epitopes are also rather conserved across different influenza virus strains. So there have been several attempts to make vaccines which mainly induce the T-cells. Unfortunately, the one which was most far in development, which even went into a phase 3 trial, turned out to not really be able to improve protection from influenza virus infection. However, it's also quite difficult to prove really the additional benefit of T-cells in such experiments. That will be a challenge also for other developers who focus on T-cell inducing vaccines.
Due to the fact that both influenza and COVID disease are both respiratory diseases, would it make sense to make a combination vaccine?
Indeed, influenza and coronavirus infect the same tissues, the upper respiratory tract in the beginning and then travel on to the lower respiratory tract eventually. And there have been clinical trials in order to find out whether you can combine the current influenza vaccines with the current coronavirus vaccines, whether giving them together would hamper induction of immunity to either of them. That was not the case. So it's safe and it is also effective to give the 2 vaccines together. There is one interesting approach for nasal coronavirus vaccine. That is a nasal vaccine which is based on influenza virus, which is made non-replicative. So this vaccine consists of an influenza virus which encodes the spike protein of the coronavirus. And when people are immunized, or when in a preclinical model animals are immunized with this vaccine, immune responses to both the influenza virus and the coronavirus are raised. Whether that will be really valid and effective approach for the future remains to be seen, but in any case it's quite interesting.
In view of the multitude of all the respiratory diseases, would it make sense to make a vaccine against all of them?
Well, there are about at least 200 different viruses only, which can cause typical common cold symptoms, respiratory symptoms. And I don't think that it's feasible to make vaccines against all of them. And I also don't think that it's desirable to do that. I think that some of these infections are important to keep our immune system, our innate immune system, but possibly also our adaptive immune system, at a kind of elevated state, so that once we encounter a real more virulent virus, our immune system responds in an adequate way.
So what do you think about new approaches to generate non-replicating inert intranasal vaccines, like the vaccine which is developed by Intravac, the AVAC10, which is non-replicating. It's based on an outer-membrane vesicle, which is given intranasally. What do you think about that approach?
What I like about the approach is that the technology makes use of bacterial outer membrane vesicles, which can trigger innate immune response and which can, as such, work as a kind of adjuvant. So that's certainly an interesting approach. I would hope that it works in the human context. It has been proven to work very well in animal models already, but very often the translation to the human system is quite difficult, because also the way of administration differs between animals and humans.
Thank you for watching this video of the series Behind the Scenes Making an Intranasal Vaccine. Please join us the next time when we continue this series.